Welcome back. From the earlier video, now it must have become clear that the phenotypic ratio and the genotypic ratio. What was the genotypic and what was the phenotypic ratio? Here at present, I am talking about the genotypic ratio. Just now, I have discussed about calculation, that is, it can be expressed in mathematical form. Is it right? Now here I'm talking for the same thing here. That is, it can be calculated. The ratios can be calculated by using mathematical probability. By looking at the phenotype of the dominant trait. Dominant trait का क्या देखना है? Phenotype देखना है. And it is not possible to know the genotype composition. Only by seeing externally we can know the phenotype ratio. But the genotype ratio, we cannot say anything about it. We cannot predict that. So that means here, how to determine that? Or how to come to know about that. So that's why, in order to determine the genotype of the tall plant, which are produced at F2, let us say. Because one case was there where both the genes are found to be dominant, and two cases are there where it is found to have one dominant and one recessive. Now, how to determine then? Only by seeing the tallness or tall plant, we cannot say that which plant is having both the genes dominant and which plants are having one dominant and one recessive gene. So, in order to know that, Mendel proceeded further. Mendel crossed the tall plant of F2 with the dwarf plant. What he did? He crossed the tall plant of the F2 with the dwarf plant, and he called this as a test cross. He called this as a test cross because he used this idea to know about the parent. If you consider here, what I said just now, that he crossed the tall plant of F2 with the dwarf plant. Dwarf plant means we are talking over here about the parent. We are talking about the parent here. If you just note down here, this is F1. One small, one capital and one small letter. Whereas F2 here we are talking that plant that is received here in F2 generation, 3 of them are tall. But out of that 3, 2 are having the genotype as capital TT and 2 are having capital and small t. So when he crossed this plant with the parent, recessive parent, means what? Homozygous here we are talking. Homozygous recessive parent. So as a result of that, he proceeded further in the process or in his experiment that he called as test cross. In the typical test cross, an organism, here we are talking about the pea plant, no doubt, showing a dominant phenotype and whose genotype is to be determined. Means जिसका genotype हमको पता करना है. This is crossed with the recessive parent. Instead of self-pollination or self-crossing. What is done here? Once again, test cross of an organism shows the dominant phenotype. That is showing that plant which is being utilized here, it is showing dominant phenotype. But the genotype of that plant is not known. We want to determine that. So that plant over here is being crossed with the recessive plant instead of self-crossing or self-pollinating. The progenies which are received here from such cross can be easily analyzed to predict the genotype of the organism, because one of the parent, that is the recessive parent, we know it. The other parent we do not know. But here we can determine about the genotype of that second parent. Now here the figure below The figure below here shows a test cross between two plants which is forming, one is forming violet colored flower and the other one is forming what? White colored flower. Now over here it is important to note that violet color flower is dominant, and that is why this is being represented by capital W, and the white colored flower is represented by small w because this is recessive gene. Now by using the Punnett square, let us understand here two parents are taken, let us say here, because we considered here 3:1 ratio, no, in case of the tallness of the plant. So in the first one case is obtained there capital TT 1/4. So that's why here we are taking capital W both gene, one parent, and the other parent is the recessive parent, that is homozygous recessive parent, and this one is dominant parent here, because the character that has been shown phenotypically is dominant one, which is having a violet flower, Now when it is considered by using the Punnett square. So what you can see over here in all 4 cases over here are heterozygotes. Means the zygote will contain one dominant gene and one recessive gene. That means these all plants are going to show the character of development of violet flower. All flowers will be violet. Whereas same thing when we consider here on the right hand side, the two parents, one parent is taken as homozygous recessive again, and the other parent is taken as dominant phenotype 1. Means what? It had formed a violet flower. So as a result of that, here we are using one dominant gene and the other one is recessive. In case of tallness, you remember capital TT, one capital, one small t, and the third one was small t and small t. Similar kind of observations has to be done here also, or similar kind of experiment was carried here in this case also. So what happened here in this case? What is happening here? 50% of the cases or 50% of the plants that are obtained here by using the seeds that are obtained are going to have violet flower, or otherwise are going to form violet flowers, and 50% are going to form white flowers. And this plant, the genotype of this plant or this parent was not known actually. The genotype of the recessive parent was known, but of the dominant parent the genotype was not known. So that means here we can say here that we have determined here that one of the gene is dominant and the other gene is recessive. And in this case, the first case, all the plants are going to show or form the violet flower. And as a result of that, the plant, the parent, the second parent is homozygous dominant. Now, based on whatever observation he made on the monohybrid cross, Mendel proposed two general rules in the form of principles or laws. Means after observation that he made in the monohybrid experiments, by considering different characters, he made some observations, he generalized that idea in order to consolidate his understanding, whatever he understood. The rules are generally called as what? Principles of inheritance or laws of inheritance. One of the laws that he gave was it is called as what? First law or law of dominance, and the second law that he gave is called as what? Law of segregation. Now, law of dominance, what does it state? That all the characters are controlled by discrete units which are called as factors. During his time, he had called factors because genes were not known at that time. and whatever factors are responsible for development of characters, they are available or they occur in pairs. In case of a dissimilar pair of factor, one member of the pair is going to dominate the other one. Means one of the member here in the pair of factor here is going to behave like a dominant factor and the other one will behave like a recessive factor. Then the laws of dominance here used to explain the expression of only one parental character. Means what? Over here we are talking about the law of dominance, that this is used to explain the expression of one of the parental character in case of monohybrid cross, and that also in F1, and also the expression of both the characters here in case of F2. I hope you understood here once again. In case of the monohybrid cross that we have discussed, in F1 all the plants are observed or that are found to be tall. Means only one of the parental character is expressed here. Expression of only one parental character in F1 generation. And both the parental characters are expressed in F2 generation, because over there two combinations are coming. Tall plants also are formed and dwarf plant also are formed there. And the ratio of second generation is 3:1, because 3 are tall and 1 is dwarf. The next topic is about segregation of that is law of segregation. Second law of inheritance, and that is actually called as law of segregation. I will discuss in the next video. Thank you.